Welcome to the deep dive. We take complex research, sometimes pretty dense papers, and really pull out the key insights for you. Today we're diving into, well, the intersection of machine learning and pharma discovery. Specifically, a platform that sounds like it could genuinely change how we develop antibody drugs.
Yeah, this is a really crucial area. For years, we've heard the promise, right? AI and drug design moving beyond just trial and error. But there's this persistent problem, even with amazing computational leaps. The success rate when you take a molecule from the lab dish, in vitro, and put it into
Huh.
a living system, in vivo, well, it's still pretty low. Shockingly low, actually.
Right, that translation gap, it's the big hurdle.
It really is. It's the main bottleneck holding back AI's therapeutic potential.
And that's exactly what our sources are focused on today. We're going to unpack the tech behind mBER, that's Manifold Binder Engineering and Refinement. It's an open source AI library from Manifold Bio. And they're not just talking about designing molecules, they're claiming this enables direct-to-vivo discovery. Sounds ambitious.
It does, but the sources we have are incredibly detailed. They cover the architecture, the mechanics, the math behind how they invert AlphaFold 2, which is fascinating in itself, and they back it up with validation at a scale that's just unprecedented. We're talking over 1 million unique antibody designs designed and tested. This isn't just theory.
Okay, let's set the scene then. We've seen AI get good at designing, small proteins, those new folds, but mBR is about full antibody formats, which have really strict structural rules, and then testing them at this massive scale. This feels like where the computation really meets complex biology head on.
Yeah, and to really get why Manifold's approach is different, we need to look at the traditional challenge. Think about how we used to find binders, things that stick to a target protein. Methods like phage display, yeast display. You screen huge, kind of random libraries in vitro.
Right, and you get loads of hits from that, but what's the actual problem with those hits? Why aren't they translating?
Well, you find things that bind, sure, but you have almost zero control over the really important stuff, the exact sequence where it binds on the target, the epitope, the geometry, the binding site, the paratope, and then you have stability, solubility. Is it going to stick to other things non-specifically? How easy is it to manufacture? All these things are critical for a drug.
So you end up spending years and just tons of money optimizing something that looked great in a test tube.
Exactly. Only to find out it behaves terribly or maybe even dangerously in a living thing. It's incredibly inefficient.
Okay, so it's not just about binding. It's about developability and function in a really complex environment. We have seen newer AI tools,
Yeah.
things like RF diffusion, Bindcraft. They're great at designing these novel, unconstrained mini binders, small proteins, totally new structures.
They are. And that's a significant advance, no question. But here's the key limitation for getting things to patients quickly. If you want a drug to move fast through clinical trials, you kind of need a format that the system already understands. Antibodies, those mini binders. They lack the built-in constraints, the conserved frameworks that antibodies have. Those frameworks give you predictability in manufacturing, in the regulatory process, and how they behave in the body. Designing a whole new protein type means starting from scratch on all that.
Which explains why Manifold zeroed in specifically on the antibody format, or more precisely, VHHs, single-domain antibodies.
Exactly. Antibodies are, well, they're the dominant format. Over 100 FDA-approved drugs. There's decades of knowledge baked into that format. Stability, safety profiles, it's understood. So if AI can reliably design a functional antibody, that's something you can potentially translate much faster. And VHHs are particularly interesting. They're smaller than full antibodies.
Which helps with.
Often better developability, they can be more stable, and crucially, they might penetrate tissues better, which is key for some tricky delivery challenges. So Manifold picked a very clinically relevant scaffold to focus on.
And this leads us to their core idea, ML2, machine learning with multiplexed libraries. That phrasing seems very deliberate. What's the strategy there?
ML2 is really about integrating the whole process. It's the AI design part that's mBER, combined directly with massively parallel ways to synthesize the molecules and, importantly, measure what they do. The big difference is they're not just designing computationally better molecules on paper. They're generating and screening potentially millions of designs at scale directly in systems that capture biological complexity, like measuring in vivo biodistribution.
Ah, okay. So the goal isn't just hitting a high score on some AI prediction metric like PLDDT or IPTM, although those are important. It's about quickly getting real-world organism-scale data on things like Where does this binder actually go in the body? Does it hit the right cells? Does it have the intended effect? That's the high-value data the models really need to learn from, right?
Precisely. That's the ground truth. How do these millions of AI-designed molecules actually behave in a biological context? This data is then fed back to train better predictive models. It creates this self-improving loop, a flywheel for learning, as they put it. Use AI to make hypotheses at scale. Use multiplexed in vivo experiments to get the answers. Use those answers to make the AI smarter. The ultimate goal is to bridge that gap between sequence, structure, and actual biological function to eventually get to maybe zero-shot drug design.
Okay, this is where it gets really interesting technically. AlphaFold-2, ESM2, these are amazing prediction models. How on earth do you get them to design something specific, like a VHH antibody with all its constraints?
All right, that's clever. mBER builds on work from the Collab design framework, which sort of pioneered using backpropagation with AlphaFold-2 for design. What mBER essentially does is invert the network. Instead of, here's a sequence, predict the structure, it's more like, here's the structure, I want a VHH tightly bound to this specific spot on the target now, tweak the input sequence iteratively until AlphaFold-Multimer is super confident that the sequence will produce this structure.
Wow. And the kicker, according to the sources, is they managed this for antibodies without having to do massive retraining or fine-tuning of these giant base models, AF2 and ESM2. They're kind of like guiding the existing intelligence.
Exactly. They're channeling the knowledge already baked into those models using two main innovations. The first one is sequence priors using protein language models, or PLMs.
Okay, let's break down the challenge there. Designing a VHH, you need the framework parts to be stable and look like a normal antibody, right? But the binding loops, the CDRs, need to be variable and functional. Standard tools might struggle with that balance.
They often do. Generic structure-to-sequence tools might make something that folds right but looks weird, evolutionarily speaking. Maybe not developable. So, mBER brings in the big gun, ESM2. This massive language model has learned the grammar of proteins from seeing millions of natural sequences. The process starts with a VHH framework sequence where the important framework bits are fixed, often based on a stable human germline like IGHV323. But the CDRs, the loops that do the binding, are masked out.
Masked out meaning the AI doesn't know what amino acids are supposed to be there.
Right. ESM2 takes this sequence with the masked CDRs and does one calculation, a forward pass. It outputs a probability distribution, technically, logits, which they call text bias for every position in those masked CDRs.
So dex bias is basically ESM2 saying, okay, based on all the VHHs I know, here are the most natural or likely amino acids you should probably put in these binding loops.
Exactly that. And this cal-text bias isn't just a one-off suggestion. It acts as a continuous biases throughout the entire optimization process. It gently nudges the design towards sequences that are not only structurally plausible according to AlphaFold, but also look like natural, developable VHHs according to ESM2. It's a balancing act.
Okay, that handles the sequence naturalness. What's the second innovation? The structural templates. If they're using AlphaFold-Multimer to predict the complex structure, why add an external template?
It's about giving AF2-Multimer a better starting point and improving confidence. Predicting the structure of a totally new antibody binding to an antigen from scratch is still really hard, even for AF2-Multimer. The search space is huge. By providing an informative structural template, mBER biases the network towards predicting a high-confidence VHH fold right from the beginning. It helps the whole process converge reliably.
And how do they make this template? Is it just a generic VHH shape?
They use a specialized model, Antibody Design 2, NB2, which is specifically trained to fold VHH structures accurately. They use NB2 to generate a reference structure for the VHH part. Then, they computationally merge this VHH structure with the structure of the target protein or the relevant part of it. This combined PDB file becomes the template fed into AF2-Multimer during design and evaluation.
But wait, if you feed it a template of the VHH already bound to the target, aren't you just telling it the answer? How does it design something new?
Good question. That's the clever masking part again. Within the template information given to AF2-Multimer, they specifically mask out or hide crucial details about the interface. They mask the predicted contacts between the two proteins, and critically, the structure of the CDR loops themselves within the template features. So the template guides the overall VHH framework fold, ensuring it maintains its integrity, but the AI still has to figure out how the CDRs should shape themselves to actually bind the target, guided purely by minimizing the structural prediction error from AF2. It's pretty sophisticated engineering. Right, so we've got the key ingredients: inverted AF2, ESM2 sequence bias, and the structural template. Let's walk through the actual four-step pipeline mBER uses.
Okay, step 1. Input prep and truncation. Starts with the target protein and deciding where to aim the binder.
Correct. You need the target structure, maybe from UniProt or the PDB, and that partially masked VHH sequence we talked about. Since Manifold is often interested in cell surface proteins for delivery, these targets can be big, complex molecules. Designing against the whole thing is computationally expensive and maybe unnecessary.
So they need to focus the AI's attention, kind of like putting blinders on it.
Exactly. First, they usually chop off the parts inside the cell or in the membrane. They use a tool called T-embed to find those transmembrane bits and just keep the extracellular domain, the part sticking outside the cell. Then, epitope selection. They find residues on the surface using a metric called SASA solvent accessible surface area. Makes sense, right? You want to bind to something exposed. They pick a random residue in an exposed region as the hotspot.
Okay, hotspot selected. Then they trim the structure down around it.
Yep, this is really key for efficiency. Using dynamic programming, they basically carve out a chunk of the target structure, keeping only the residues within about 25 angstroms of that chosen hotspot. This drastically reduces the size of the problem for AlphaFold-Multimer, saving compute time while still keeping the local environment of the binding site intact.
Makes sense. Then step 2, template prep. This is where the sequence and structure priors we discussed come together.
Right. ESM2 calculates those sequence bias logits. Then, they actually sample one sequence based on those probabilities and fold that sequence using Nanobody Builder 2.
Yeah.
That gives them the specific VHH structural template for this design attempt. They merge that VHH template with the truncated target structure template into one PDB file. That's the combined structural prior ready for the main event.
Which is step 3, iterative optimization via backpropagation. This is the core loop where AF2's feedback shapes the sequence.
Exactly. It uses a method from Collab Design called Design 3-Stage. The system repeatedly asks AF2-Multimer to predict the structure of the current sequence bound to the target, calculates how far off that prediction is from the ideal bound state, low error, and uses that error signal to update the sequence logits, pushing them towards a better solution.
Okay, let's get into the math slightly because the equation they use to combine the design pressure and the sequence bias seems really specific. It defines the total logits vector. Tell them about it.
Yeah, that combination is central. The total logits vector, VisWell, is defined as VisWell plus VisWell delay.
So Vislit's design is what AF2 is trying to change based on the structure, pushing it towards binding.
Correct. That's the part being actively optimized by the structural loss. And Vislit is that constant guidance from ESM2, saying stay natural. Tisladol is a temperature factor that controls how strongly that ESM2 bias pulls. Low test bias means strong pull towards natural sequences. High test bias gives AF2 more freedom to explore weirder stuff. Manifold tunes this carefully.
Okay, so that's the combined direction. How does that translate into the actual sequence vector that AF2 uses at each step? There's another equation for that.
Right, the final sequence input vector is calculated using this 1 lambda plus softmax.
Looks complicated. What are lambda dolas doing there?
They manage the optimization process, kind of like annealing. Lambda is a parameter that increases over the optimization steps. Early on, lambda is small, so the fatters is closer to the raw combined logits for lost men, allowing for more exploration based on the raw scores. As optimization progresses, lambda gets bigger, so the softmax term dominates. Softmax converts logits into probabilities, sharpening the choices.
So it starts exploring broadly, then gradually focuses in on the best options. And Tex is at another temperature.
Yeah, TECAS is a temperature for the softmax part, and it typically decreases over the optimization steps. High temperature early on makes the softmax output flatter, meaning more amino acids have a chance, again encouraging exploration. As TECI drops, the softmax becomes peakier, focusing probability on the single best amino acid for each position according to the combined signal.
Wow, okay. That's really elegant. They've engineered the math to let it explore, but then force it to converge on something that meets both the structural needs from AF2 and the naturalness needs from ESM2. What's the main structural loss it's trying to minimize?
The primary driver is the predicted aligned error, or PAE, specifically the PAE between the binder chain and the target chain. PAE is great because it measures AF2's confidence in the relative positions of different parts of the structure. Low interchain PAE means AF2 is confident it knows how the binder sits on the target. They use other metrics too, but minimizing that interface, PAE, is key for designing binding.
Okay, optimization done. That brings us to step 4, evaluation and scoring. Gotta check if the design actually worked, computationally at least.
Right. They take the final design sequence and refold it using a different set of AF2 multimodal weights. This is important for avoiding bias. like asking the same teacher who taught the class to grade the exam. The key score they rely on here is IPTM, the Interface Predicted Template Modeling Score.
Why IPTM specifically? We hear a lot about PLDDT for single protein folding quality.
It's a crucial distinction for drug design. PLDDT measures how well the individual VHH domain is predicted to fold on its own. And Manifold found that while high PLDDT might correlate a tiny bit with off-target binding, maybe just general stickiness, it wasn't a good predictor of on-target, specific binding success.
So a perfectly folded protein isn't necessarily a good binder. It might just be generically stable or sticky.
Exactly. Sometimes really stable proteins expose patches that just glom onto things non-specifically. That's bad for a drug. IPTM, on the other hand, specifically measures the predicted accuracy of the interface between the binder and the target. Manifold showed it correlates much better with actual experimental binding success. So filtering designs based on IPTM enriches for functional, specific binders, not just stable blogs.
Okay, the engine sounds solid, but the validation, that's where the multiplex library as part of ML2 really comes in. You said over 1 million designs tested. That number is just staggering.
It really is transformative. This is where the platform approach shines, linking design directly to large-scale experimental data. The numbers: 1,153,241 unique VHH binders designed. They targeted 436 different human cell surface proteins computationally. Then, experimentally, they screened these libraries against 145 of those targets using their high-throughput platform.
And the number of interactions measured.
Over 100 million protein interactions in one go. Just to put that in perspective, think about the PDB, the Protein Data Bank. Decades of work by scientists worldwide, maybe 200,000 structures total. Manifold is generating specific interaction data on an industrial scale.
That's the real differentiator then. It's not just the design, it's generating this massive labeled data set linking sequence to function. What were the success rates like?
Overall, pretty impressive. They got specific, significant binding hits against 65 of the 145 targets they tested. That's a 45% success rate at the target level.
45%, compared to traditional methods where you might screen millions of compounds and find maybe a handful of starting points after a ton of work. That's huge.
It's a massive leap in efficiency, yeah. But it's important to note, the raw hit rate per design before filtering was actually quite low. Median was around 0.4%. Which tells you something important.
That the computational filtering is absolutely essential.
Absolutely critical. That APTM score we talked about, when they applied filters based on it, say, only taking designs with APTM above 0.7 or 0.8, the effective hit rates jumped dramatically into double-digit percentages. And for the best, most designable epitopes, those filtered hit rates hit 38% to 40%. That's right up there with best state-of-the-art, often closed-source models.
And crucially, this filtering proves IPTM is picking out specific binders, right? Not just sticky ones.
Exactly. They looked at the off-design hits binders sticking to targets they weren't designed for. Those rates stayed low and flat even when they filtered for high IPTM. Meanwhile, the on-design hits increased sharply with higher IPTM. They saw something like 2,200 on-design hits versus only about 76 normalized off-design hits. It strongly confirms IPTM selects for the intended interaction.
The results also revealed something tricky, though. This idea of epitope sensitivity. Success wasn't uniform across a target protein.
Yeah, this is a really important finding and a current limitation. Experimental success turned out to be highly dependent on which specific epitope they targeted. They used PDL1 as an example. They designed binders targeting several different spots, different hotspots on PDL1. But almost all the binders that actually worked in the experiments ended up binding to just one specific site on the protein.
So even if the AI designed it for spot B, the successful binders all went to spot A.
Pretty much. It suggests that while the tech can target a protein and even a region, it can't yet guarantee success for any arbitrary spot on that protein surface. Some epitopes are just much harder to design binders for than others.
Why might that be?
It's still an open question. Could be the flexibility of that part of the protein. Maybe the VHH scaffold just can't easily make the shape needed to bind there without becoming unstable. We don't fully know. But the takeaway is that each epitope almost acts like a completely separate design challenge. The next step is figuring out why some are non-permissive and teaching the AI how to recognize or overcome that.
And that massive all-against-all screen, the 100 million interactions, that gave them tons of data on off-target binding, too, which must be incredibly valuable.
Gold standard data for developability? Yes. They found about 36,000 off-target hits total. Sounds like a lot, but most came from a few stinky designs hitting many things. Having this data at scale lets them identify and flag potentially problematic designs or scaffolds much earlier. It's crucial for predicting safety down the line.
Okay, so the mBER engine designs, the ML2 platform validates at scale, it works. What's the big picture? What is Manifold Bio actually doing with this capability? You mentioned tackling grand challenges.
Their big, publicly stated focus is on solving really tough drug delivery problems. The prime example is getting drugs across the blood-brain barrier, the BBB. This has been a huge roadblock in medicine for decades. Very few molecules get into the brain effectively. The traditional approach is usually to piggyback onto one known transport system, like the transferrin receptor, TfR.
Right, trying to engineer a shuttle using TfR, but the sources hinted there might be many other ways in. Hundreds of potential portals.
Exactly. And that's where Manifold's platform is uniquely powerful. Instead of spending years trying to perfect just TfR binders, mBER can generate huge libraries targeting hundreds of different potential receptors on the BBB simultaneously. Then, their ML2 platform screens these in vivo to see which ones actually get payloads into the brain tissue. It's a systematic, massive search of the biological possibilities rather than betting on just one horse. It's a totally different way to approach delivery discovery.
And the result of all this screening and data generation is this proprietary asset they call the epitome. What exactly is that?
The epitome sounds like their internal map. It links specific binding sites, the epitopes on these surface proteins, to the actual in vivo biodistribution data they measure. So it's not just a catalog of binders, it's a functional atlas saying, bind this spot on this protein, and the molecule tends to go to this tissue or these cells. It's a toolbox of validated delivery modules. Their ambition is huge to build this map for potentially all tissues and cell types, not just the brain, solving delivery systematically.
Okay, but it's not perfect yet. Let's talk limitations. What are the big hurdles they still need to overcome?
Well, the first one we already touched on, the epitope constraint. They still can't reliably hit every spot on every target. That means wasted effort computationally and experimentally. Understanding why certain epitopes fail is key. Is it protein dynamics? Steric clashes. If they can figure that out, they can perhaps predict designable epitopes better or even engineer solutions. That requires more mechanistic understanding.
And the other big one is probably cost, right? Running these massive AI models isn't cheap.
Definitely. Computational cost. Getting those really high IPTM scores, which correlate with success, often requires running more design simulations, deeper sampling, and that costs time and money. Future work probably needs to focus on making the design process itself more efficient. Maybe better loss functions, smarter ways to manage those parameters like lambda and temperature, or maybe entirely new, faster folding models down the line.
But even with those challenges, the most profound impact here might be the data itself, this virtuous cycle of data they're creating.
Absolutely. I think this is the long-term strategic advantage. Manifold is generating millions of labeled data points. Each point connects an AI design sequence and structure to real in vivo functional data. Public datasets often lack that direct biological validation at this scale. This massive high-quality corpus, over 100 million interactions and counting, is exactly the kind of data needed to train the next generation of protein AI models.
So mBER is powerful now, but the data it generates is potentially fueling the successor to AlphaFold-2 or AlphaFold-3 or whatever comes next.
That's the flywheel concept, yeah. Better models, trained on Manifold's rich, functionally validated data, will lead to even better VHH designs. Those better designs will have higher success rates in the ML2 platform experiments. That generates even more higher quality data. It's a self-accelerating loop. And that cycle, they believe, is the path towards truly programmable medicine, where you can specify a desired therapeutic outcome, and the AI can design the molecule to achieve it, maybe even zero shot.
So recapping. We've looked at mBER, this clever combination of inverting AlphaFold, using ESM2 for sequence priors, adding structural templates, all to design specific antibody formats, and crucially coupling that with the ML2 platform for massive in vivo validation. It's really about building this map from computational design to actual therapeutic effect in living systems. A foundational step, especially for tackling things like targeted delivery.
Yeah, the work really underscores a shift. Maybe the biggest bottleneck isn't purely designing molecules anymore. We can generate millions computationally. The real challenge, the real value, is in generating the biologically relevant data about how those designs behave in vivo at scale. So here's a thought to leave you with. If the future really is specifying, say, deliver this payload to this specific type of neuron in the brain, what piece of biological information that we don't currently capture well in our models or databases will AI need next? Maybe it's about the dynamics of receptor interactions on the cell surface in real time or local tissue microenvironments. What's the next layer of biological complexity AI needs to master for true zero-shot design?
That's a deep question. Where does the next layer of necessary biological insight come from? Definitely something to chew on. Thank you for joining us for this deep dive.